Good morning. Can you hear me? Okay, perfect. Welcome, everyone. And I'm happy to see all the faces, and some probably new. Some is we know before from the past conference. So, and I'm happy to see all of you come here, and we're going to spend about 4 hours together, go through this workshop. So in the past, almost past decades, we focus on metabolomics, and I feel most of you had already had experience with MetaboAnalyst, right? Raise your hand, please. Yeah. So yeah, I do not want to repeat too many things with MetaboAnalyst itself. I will introduce some of the key features and also like a raw spectrum processing, which we receive a lot of questions. But for today, and we are going to focus on more application of metabolomics together with microbiome for kind of application integration. So I feel this is probably more meaningful. But on the other hand, throughout the workshop, if you are not too much into microbiome, you want to continue with metabolomics, MetaboAnalyst is all fine. We are happy to answer your questions. So, so this is our schedule for today, and we will have an overall introduction because all the omics disregarding microbiome, metabolomics, or RNA-seq, even multi-omics, they share some strong patterns, and it's actually relatively easy to follow conceptually. The difficult part is actually do it practically, and so that's why the reason we developing tools to help you. Once you know the high-level concept, actually doing it shouldn't be that hard, and you do not have to learn our programming language, which is very powerful. But if your background is biology, and it's really distracting for you to keep up with the programming part. So that's I found out is very important. We need to know the concept of omics data process in general. Then we are going to focus on metabolomics, and this time we just focus on MS1, MS2, raw data processing. Stats we will not cover, but we do have Nature Protocol 2023, and we're going to have a new Nature Protocol maybe later this year. So we are continue covering the stats, but not for this workshop. But we do have very detailed protocols you can follow either before or the coming, okay? So the key idea actually in central is microbiome. Microbiome is very complex. We need to shift gear from understanding metabolomics to actually also incorporate microbiome. And microbiome, if we're talking in the past decades, they are really doing sequencing based in the marker genes or shotgun sequencing, and we understand who is there, like microbes and their genes, and how we integrate with metabolomics or metabolites. So this part is we would like to discuss in the last one as microbiome metabolomics. But before we get in there, we need to do a very good job with microbiome data analysis itself. So we introduced MicrobiomeAnalyst, which actually we developed almost 10 years, and is widely used. I'm not sure people in metabolomics society are aware of that, but it's very similar to MetaboAnalyst and widely used. So I would just hope if you are doing microbiome metabolomics, and very, very easy. And so we need to just get some concept through, and then you can click buttons, upload your data to get it done. So this one I'm going to pause and just follow the GitHub link. You can download the slides for today, and it's all in one slide, 116 pages PDF. So I'm going to have 4 sessions. Yeah, like each topic, a demo. Yes? I asked just the free Wi-Fi, no password.
It's not really working, but anyway.
I realize that. I talked to people in back outside. They are addressing it. I'm not sure how fast they can addressing, but if too much, if it's too much stress on the bandwidth, I thought 5G will is here. But yeah, we can always follow the slides and mentally go through it, ask questions in case you cannot follow. So this is something I cannot control too much, but we have a lot of slides and we can just mentally go through it, no problem. So yeah, I'm just keeping here. This is GitHub, and my lab with Metabolomics 2025. In between is underscore, and download that slides, PDF, and we just can just go through it together, even you cannot follow if the bandwidth is too slow, and so. So if so, you cannot even log in to download, or just slow. You can still log in. Yeah, so welcome everyone. And so I'm just thinking in the worst case, you cannot use your cell phone, use a different network to download the slides. So at least we can learn through the slides, screenshot, even you cannot follow real time. But yeah. Take a photo. So again, welcome everyone. I know a lot of people join, and we do know the bandwidth is not fast to accommodate everyone to follow us to hands-on practice, because you need to access our website and to follow what we are doing, because the bandwidth we cannot, maybe you cannot follow, but please download the slides. We can just go through it mentally. Maybe some of you will be able to follow later. And so this is the current situation. If you just download it, if you cannot go Wi-Fi, maybe use your cell phone network, just download that PDF slides of 116 pages. At least you can just go through, and we can learn together. And that's the best one. I'm just thinking how we can go through. So we also have, this is a GitHub. All the data, slides, and material is in a GitHub. And we have the tools website here, but that's why I'm just not sure you can click to get there, because bottleneck is here. Our tool can support several hundred people simultaneously. So this part is, but if here you have the bottleneck, there's so much we can do. And later you can do the omics forum. So I believe omics forum and all of the online, probably too slow. But I just do hope you get these slides so we can just go through it. Okay. Okay, let's move on. So today's topic is actually application of metabolomics to study microbiome. And metabolomics play a key role in microbiome functions. And here's we show that fecal metabolomics is a functional readout of all these interactions, host, gut microbiome, and a lot of the novel discoveries, and it just even is together with the diet. So there's a lot of complexity there and a lot of discoveries. And you can see it's very routine. We have the sequencing, we have metabolomics, and we have all the cool data, and how do we make most out of it? And here's this is today we need to just focus on this. And basic levels, we need to do the best of metabolomic data analysis. We need to do the best of microbiome data analysis. Then we try to integrate. So there's no magic bullet, but it's very practical, and we just do step by step and the integration, and there's concept and there's tools. So the reason motivated me into this field is this stark contrast between taxonomy or composition or microbiome and their actual function, because the microbes mainly function through their metabolic capacity. So they are very different. So sequencing microbiome, and I think that's the whole picture is far from it. And everybody see this thing and realize we need to do sequencing, we need to do metabolomics. So this is part the whole community kind of have the consensus. And we need to know who is there by sequencing them, by find out what microbe is there. Then we need to know basically their functional capability, what can they do. And now we maybe do from marker genes to shotgun metagenomics to get all the gene content there. Then eventually we know what they are doing. So we need to do this proteomics or metabolomics. So this is very consensus. If we do the microbiome study, and I see majority, we do metabolomics plus sequencing. So today we are focusing on the marker gene, shotgun metagenomics, and metabolomics, and how we integrate them. So with this question in mind, we have the 3 tools, and MetaboAnalyst is what are they doing? Basically, we want to do this metabolomics data processing, functional analysis. And who is there is doing a marker gene. We are doing this 16S or 18S or ITS. And we use MicrobiomeAnalyst to actually profiling it, understand it, and compare, get a signature. And eventually we want to see their functions. So MicrobiomeAnalyst can give you functions, but we also recently working on system biology using genome scale metabolic models. We call it MicrobiomeNet, and we can try to understand the statistical association, their metabolites, what they indicate, how they interact, and metabolic they compete or complement. And this is when this is our current focus. So we hope by integrating these 3 tools, we're able to get quite a decent level of understanding. So that's a big picture. I'm just moving on to omics data science. I mentioned before, omics data is everywhere, but it's become very easy nowadays conceptually. So let's conceptually feel it's not a thing that we are stressed about, we are scared about. It's really something, it should be a routine, okay? So let's just overcome this fear. If we think omics data is complex, we just really need to conceptually see how it's been generated. We need to have high throughput technology. We get this data table, and from the data table, we want to understand what they are doing. This is really, all the omics follow this. And if we talk about the omics technology, and we only need to focus on 2 omics technology mostly, and I know there's other things. One is called NGS omics, is sequencing, okay? Today we're talking about marker gene, shotgun, all based on NGS technology. If we move to the other one, is MS omics, mass spec, and proteomics, metabolomics, exposomics. If you're not quite sure about exposomics, everything, environment, food, drug, microbiome, pollutant, everything is almost part of exposome. So they all use mass spec-based approaches. So from sequencing, mass spec, we can almost get a majority of this omics data covered. And so this is conceptually, it seems diverse, but it's very unified also. So if we deal with this omics data, we need to have also think about 2 things. One is raw data processing, which is very big and kind of slow, because the big data, raw data processing is slow. We need to be patient. We need to have relatively good computing power. But nowadays, most of data analysis is on my laptop. Laptop is so powerful, and so I do not think it's a big thing. And you also have access to cloud, so it's not a big deal nowadays. and we just need to set it up for once. Then you have standardized protocol, standardized pipeline. You can run it again and again. And the challenge part we spend most time is the complexity issue. This complexity is really reflects on the table. We have the abundance table, and sometimes you can open with Excel, but you need to understand it. That is challenging. That nobody can help except yourself. And you design the study, you know the biology, and you know somehow have a vague idea about what you're looking for. So you need to back and forth with your data to feel it. And if you really like somebody else doing it, that's not going to make a great discovery. So this part is really in your hand, and you need to be familiar with the concept. You need to use the tools and back and forth until you feel satisfied. You write that paper. So the first size challenge actually is standardizable. Somebody else can do it for you. That's something. So slightly more details about what the individual doing is that the raw data processing, more or less standardized. You have it, but it's technology specific. Mass spec, NGS is different, but it's not too hard. And once you set up, it's doable. But before you do the statistical analysis, you need to understand some challenges. You need to missing values, data filtering, data normalization. This is omics specific. For example, today we're talking about microbiome and metabolomics. They use different statistical approaches to do the data processing normalization, and they are all justified in their own field because the data distribution is kind of different. We're going talking a little bit on that. So that data processing normalization is unique. But once we go to the statistical data analysis, machine learning, it's just purely data science. And really, it's a table. You can do, you can just almost anybody from computing science, statistics, you can do it. But the first two steps is really unique. And the last one is the functional analysis. That also statistical method is more or less the same, like enrichment analysis. The library is different. You need to create a functional library behind it. Metabolic pathways or microbiome kind of different function signatures, that's unique. But you just think about this. Once you understand the overall flow and you understand the uniqueness, and it's really conceptually easy. You swap the component, stuff, and replace with the new steps, and you can actually do it very well. You just always focus on biology. So go deep further, and each step processing, normalization, stats, and interpretation. And it's all more or less well established. And here I show metabolomics, okay? If you do MetaboAnalyst, all of this concept is built into that tools. We follow literally step by step. And some of the time your data does not have the missing value, so you can skip missing value estimation. And but you need to do the normalization. So you still have a lot of the things just click through if your data. But sometimes you need to stop and think and back and forth and try what's the best normalization approach, which statistical method the most suitable for my data. So this part require you attentive when you do data analysis. And again, it's if you do microbiome, we have MicrobiomeAnalyst, and you will see some of things the same, some things different, just because the uniqueness of the data distribution. But overall, the concept, the flow is the same. So, and for me, I myself is from medical background, also from biological science background. So I do feel there's not so much. The data analysis should be in the hands of the PIs, the student who designed the study, who actually focus on biology. And this is the tools we designed in the past few years. And for RNA-seq, or gene expression, metabolomics, microbiome, and we follow the same idea, same flow, and same interface thinking. So if you're familiar with MetaboAnalyst, you should be very confident. I can do RNA-seq analysis. I can do microbiome analysis. If you've never done it, you just go through the slides and think about it and use the tools. You should be able to do it. Really, omics data analysis is not that so complicated. You just need to follow this key concept. And how do we do the multi-omics? and multi-omics, and everybody more or less doing part of it. So we want to integrate multi-omics, and so we do individual omics data analysis, and we get patterns and signatures. We're wondering how they connect with each other, and we can think about it, but we have a database, like we have knowledge, have networks, and we can project this signature into the network, and we can try to see how they're linked, what the functions pathway is actually telling you. So this is more like a qualitative integration or knowledge-driven, and as long as you have this huge knowledge database, we can do integration, no problem. And so this is very intuitive, and we also have tools for that. On the right-hand side, we have this data-driven. We want to see how these different omics, are they changing together, are they changing also separate? And this is data-driven, and so we need to use some statistical, especially multivariate kernels, or dimensionality reductions, or even machine learning to detect that and visualize how they change. So this part is really dense in stats and machine learning, and we try to give it more visualization to help you. But it's data-driven, so even we do not understand too much about online biology, we can see how they covariant, how they change together. And you need to start thinking about how that making sense biologically. But this is something which I'll just tell you, two complementary approaches, knowledge-driven and maybe data-driven. You need to data matrix, which is detecting how they covariant. There's not so much more deep you need to understand, but these two concepts I think is very complementary, and we have tools for that too. So these are tools I just mentioned. We consciously build these tools, follow this concept. And so we have this raw data processing, we get the statisticals, and we found the signatures, found the patterns, then we integrate them together. And this part is really, we just need to be aware of the uniqueness of the data and use the proper tools and select the right method. And when you write your manuscript, just make sure you respect what kind of concept at the media high level. You do not need to go to very low level, and that should be enough. So that being said, a little bit advertisement is that we do have 12 weeks omics data science training. I just mentioned earlier. It's really, you need to focus on the concept and followed by the tools uniqueness. Then you should be able to really proficient doing the data analysis. If you're unhappy, you really want to do the cutting edge, you're a developer, you can really do it, but at that time, you're not, we're more talking about biologists, we're talking about clinicians. So if you want to do that, that's more than welcome. The current thing is that most of data analysis, the challenge is actually is the biology. It's not, so you use your background knowledge and use the tools back and forth, think about it to understand. So this is something I would like to, so here's, we have a summer boot camp in August, but it is in Tokyo time, if you're from Asia region, and it's the right time. But once you go to fall, it is Montreal time. So this is some things, unfortunately, European time, currently we do not have it. So either Asia time or Montreal time. So now if we're able to follow, and this is today, if we do a lot of demo, is try not to open multiple tabs, and people sometimes open multiple tabs and overwrite the session. So this part, you do not do it. And if you keep on to follow, form a group, because they will reduce the bandwidth. So you see your neighbor's screen rather than use your own screen. But I'm just not sure how feasible it is. But this is in the past, we try to reduce the bandwidth. But today is actually, I'm just really surprised to see so many people. It's the largest audience, actually. Probably, yeah. Yeah. So for today, we have example data for you to follow. If you can download and follow, it is IBD study. I think this is from the human microbiome project, phase 2. And they have pregnancy, they have IBD, they have diabetes. So they're collecting both microbiome sequence data and LCMS data to try to understand IBD. And they published several papers. And we actually use this as a benchmark for our study. And we can see we can get the same or better result. And So this is a little bit background, and so this is how the metabolomics being carried on. And the one thing that they only did LCMS 1, they don't do MS2. So if we do a demo later, MS2, we use another example just show what how MS2 can be done. But this study they only have MS1, and so it's really no worry. You can just follow what's being shown here and go through the slides, watch the demo, and we can just slow and ask. I will be happy to answer your question if you're not able to follow. And so overall, that we're going to do metabolomics. We use MetaboAnalyst.ca to do raw data processing and picking, profiling, and we can do some function or even stats analysis. But if it's not, and you just watch what we have done, and you can do it later when you return in your hotel or after you return to your home, your university. And microbiome, we are going to MicrobiomeAnalyst 2.0, and we're doing a taxonomy profiling, and mainly focus on marker gene. So if you have shotgun, no problem for that, but marker gene usually is the most commonly one. And multi-omics, we're just using microbiome metabolomics. We use MicrobiomeAnalyst and MicrobiomeNet to do the integration and interpretation. So we slightly behind the schedule, but just because we are waiting, so we should be able to catch up, and no problem with that. And now we move to metabolomics data processing. At the moment, if you have questions, raise your hand. I'm happy to answer. No? Okay. Metabolomics and main focus on untargeted LCMS metabolomics. Because if we're doing targeted, we get a concentration table, and understand the concentration table is relatively straightforward. And we have done it like 10 years ago. Now it's getting much better because we have large coverage of we can get thousands of compounds being identified and profiled. But if we're doing an LCMS untargeted, and this is we have more challenging. We have the LCMS 1, and we also can do MS2. So MS2, we do the DDA or DIA. DDA is we select the peaks that's most abundant, and doing MS2 fragmentation. And DIA is we just have overlapping windows. So all the peaks we select for fragmentation. So all of this will make the data larger, more complex. And how do we do that? I just mentioned the MetaboAnalyst, or we are working for supporting for both targeted and untargeted. So for untargeted is we just process the data, we found the patterns, then we try to go back to identify the peaks and relate to the major functions or signatures. So this is really for discovery, but if you're using a kit, and here you just get a concentration table. So more or less, we focus on the untargeted just because this side is relatively straightforward. And MetaboAnalyst, you guys already use it, and we have done, this is version 6 we published last year. And so we focus on untargeted metabolomics just because it still remains challenging. And we have done LCMS 1 and recently MS2. We continue adding more functions. And we actually more supporting now is to focus on exposomics because exposomics seems a lot of the new things and separate from metabolomics. So this is the interface you should be familiar with. On the top, this is all untargeted. On the center is all basically, this is a statistic method. So with this statistical method, it doesn't really matter. You can be you just a table, be it peaks, be it compounds, doesn't really matter. So the center row is just a generic table you upload doing data. The bottom is compound names or compound table is more targeted. So you need to upload a table with the IDs. And so I mentioned earlier is that we would like to use these tools to streamline and simplify the data analysis. We do not want to make it complicated unless it has to be in that way. So the overall that you know how to drive, but you do not have to drive. So you just let it more or less let it go, but you do have the power to overwrite. So this part is that we think with the more recent AI, and we can do more customization stuff. But it's really should be as intuitive as possible, as long as we are aware of the concept behind it. So you cannot just have a black box, and here's input, here's output. And we really expose the steps, but you do not have to just figure out how exactly do this, do that, but you need to be aware every step. We've been transparent. And so the education and learning should be there, but actually very practical that writing the code is not unnecessary. So for the protocol, we designed based MetaboAnalyst what the protocol we have in mind if you're doing untargeted metabolomics. So this is something we follow some of the study like 2011, maybe 2012. The field of metabolomics, what they try to do is LCMS1. Use this for quantitative analysis. And see, we give a simple example. We have 10 control, 10 disease. Now we also need to have pooled QC. So pooled QC is adequate of each sample and have a pooled one. And we can also have blanks, basically no sample, just a solvent. And this is things you need to more or less follow. This is most kind of used. Then for the LCMS1, we're running for each sample, doing quantitative analysis. But for MS2, we mainly just doing a pooled sample, because you do not need to do the identification for each sample. You just do identification on the aggregated sample. But we have, let's see, we have 3 replicates. We can do the consensus. So overall, it's try to minimize the analytical things, but also maximize how you can gain. So this is something. On the bottom, we need to collect this spectra, process them, and get a table. And clearly, this table can have, let's say, 20%, we can annotate them. Another 18%, we still have peaks like retention time, mass. But all the table, we can do data analysis and statistical analysis and do functional analysis. So this is, if you are doing targeted stuff, you do not do the first step. You're just starting from this table and doing the stats, doing the function. So this is why I think untargeted is slightly more complex, but overall it's doable, and it's getting easier and easier. And so this is the LCMS spectral processing overview. And this is the main bottleneck actually, but it's not what we have too much control. And you need to convert your spectra to open format, MZML format, and use a tool called Proteo Wizard. It can do both LCMS1 or MS1, MS2. So this part is, I get a lot of people asking how they can do that. So we are going to a demo after after a few more slides. But overall, that it is well established, and you can talk to this Proteo Wizard community how hopefully they can continue move on, get it better. And if we convert it, we can do the LCMS1 processing using MetaboAnalyst R, which we make it automated and more optimized than the other tool called Asari, which is complement to what we are doing. Its result will be different because it's based on different concept, but it's very useful, a complementary approach. So MS2 is really, we can do DDA and DIA in a consistent way. I'm not aware other tools can do the same like what we are doing right now. And we can do functional analysis, Mamitrog and GSEA or MSEA, metabolite asset enrichment analysis. So this is under the hood as we do MS1, MS2. MS1, we do the optimization of the parameter because peak-peaking detection need optimization, but you do not need to manually do that. The program do it for you based on some criteria, which is most people feel it's reasonable. So And so this is optimization and get to the table. And MS2, we need to do the peak deconvolution, because once we do the high resolution, high throughput, complex biofluid, we pick peaks, and they do have some different things mixed together. We need to deconvolute. So if you do a single cell analysis, and this deconvolution is very common, because we really want to make sure that clear from parent ions and fragment all linked together. And with MS1 and MS2, we search a large database. We can identify them, and then we'll update the table and doing the functional analysis. So this is the things we show about how to optimize the parameter. And some people saying, they're not necessarily optimal all the time. So that's true. We try to create this quality score, try to balance different criteria. So we spend our time, and we do not think there's always the best one. But overall, it's better than you don't do it. We can always improve, I think, 5 to 10%, sometimes higher. So it's just actually about 8 minutes, so we optimize parameter.
Can we interrupt for a question? Or?
Yes.
With regard to DIA data, do you know how to create pseudo DDA data out of DIA? Like trying to deconvolve DIA.
Yeah.
Right. So this is something that might be covered at some point in your tools. Maybe not today, but I'm curious about this.
Yeah, we're going to have slides on that very soon. Okay. Yeah. Thank you. Yeah. So far it's mainly focused on MS1, but we're going to MS2 very soon on that. So with optimization, we can see we identify more peaks, slightly more compared to the default standard wave, which is XMS. So our peak picking is based on XMS, which is well established and actually most time perform quite decent. But just performance parameter optimization takes something, and we're just adding about 8 minutes. We can get a better result. You can see, more real peaks. Okay, and this is consistent. We just show you one, but overall, it's if you trust yourself, you can always manually use the default or tune yourself, or just let the system figure out. And also, after you do this, the quantification is better. So we do the serial dilutions. We actually get correlation with the known ratio is much better. And more biological variance means if we do the compare different groups, and we want to see the PC1, PC2, PC3 capture more variance, which means probably that's what you would like. So after doing optimization, indeed you have like 5 to 10% more variance being captured in the top 3 PCs. So we think that's useful. Of course, you can visualize, see whether separation better. Most time it is. So after doing optimization, you have better separation. And this is another one called Asari. And this is my dear friends, Dr. Shuzhao Li from Jackson Laboratory. And they publish Asari. Python-based, so this is quite cool, and because we're doing R, and it's Python-based and a very beautiful software engineer, and what they try to do is using high-resolution mass spec, they try to align from the mass peaks first, not from retention time, and then have the consensus mass check. And you can see some of the signal you're not quite sure, because you need to waiting for the Gaussian peaks, but here they just require consistent signals there. Then across different samples, the signal will be enhanced. So disregarding, once you have more signals, the data point, it became more Gaussian-like. And so this is quite intuitive. Most important thing for us is it's fast. Memory CPU is actually much lower compared to XMS. So this is something we just let you know if you want to try. And there's another option for Asari, either command line you can download or just via MetaboAnalyst. So that was MS1. Now we're doing MS2, and we have DDA and DIA, and this is really DDA, DIA, both do peak deconvolution and the searching database, and with annotation. So it's very, very traditional. We are not claiming something significantly different, but we just want to make it smooth and default performance is very good. So let's just go through it quickly. So DDA, what's new? So XCMS do support DDA, but I just mentioned about their it's basic support. What recent finding is DDA do have this contamination. So you can, when you select this even narrow window, and more than half of time you select multiple ions actually, and this is reported by the Gary Petty's group, and we confirm ourselves. So we need to deconvolute them before we do the assignment. And so how do we do that is actually follow this one. You search database, identify with potential, how you can interpret this MS2 patterns combined. And we also use this reaction networks to try to, if we couldn't find it in database, can we expand, try to explain them? So overall is we have this deconvolution algorithm actually working very well, and again, it's about 5 to 10% compared to not deconvoluted. We are talking about a complex biofluid like plant and fecal samples, blood, urine. And if you're very simple, probably you don't see it because you do not have contamination. And this is DIA you mentioned. And the DIA approach, we use deco-method DIA. So this is from my another colleague, Jianguo, who developed this R package called deco-method DIA. And so it's in theory working very well. In theory, it's actually very solve the problem. Practically, it's just slow, and we're running for several hours, sometimes days to do it. So we wrote it in C++, so we can just get it done in 20 minutes. Should be within 1 hour. So this is we are not going to talking too much, but this is hopefully answer your questions. And so, yeah, this is a deco-method DIA, so it's actually very comprehensive and solving a lot of the problems. So all we need is just make it building into our pipeline, make it fast to deconvolute, and from here is we can just do the annotation and stuff. So if we do the annotation, we need to have a database. And we do not collect in our database like Matlin or stuff. We just compile all the public database available. So you can see this public database. We have about 10 million spectra, about 1.5 million unique compounds being covered. It's total about 7.2 gigabytes. And it's biological, like functional biology and exposome and lipid. So it's separated. You can use the complete library by default. Our search algorithm is very fast. You can and do the complete, but sometimes you have complete may introduce some false positives. So you probably just choose the most relevant database. And you can download it from MetaboAnalyst website, the whole there. And you can add your own database. This is a schema, it's a SQLite, and once you add in your own in-house database, you can actually have much more confident ID because you have your own retention time and based on your own column. Otherwise, the identification, we do not consider too much about the retention time match just because it is not comparable, but we do use retention time, maybe just discriminate. This is not the same peaks or stuff. But in your case, this is more meaningful. So we did some benchmarking about DDA, DIA, and you can see this deconvolution on bottom two rows, okay? Bottom two rows. The bold MetaboAnalyst, we have this deconvolution. So the last row is non-deco, is not deconvolution. So you can see about 10 minutes, but you improve about 3 or 4%. So this is something have slightly more peaks more compounds being identified just because you increase the quality and stuff. This is more, the performance much better the more complex your biofluid became. And this is a standard mixture, so 400 some compounds, not that complex. Once you go to thousands, then its improvement became better. And this is DIA, and again, is that we try our best to use other tools like MS Finder and XCMS is a series. Somehow they either taken too long or sometimes too short, but when you take too short, they don't give you a result. and we are not quite sure what's going on. So we blame ourselves, but we try our best. We do not know what's going on with this data or with the tools at the moment. Our tools can get a result more or less decent. And again, if we use a complete database, which is 20 million spectra, if we don't search it, it's, for this one, yeah. You get some time, but it's not that prohibitively long, so it's all fine. Just go to see the bottom things. And this is very typical. And different tools, different pipelines get very different result. So this is something everybody be aware, and we are all surprised every time, and we try to reduce it, but it seems it stays, and we do not have very satisfactory explanation why things like that. And but it seems if you're looking for the functions, robust signals, they more or less agree, but just peak level still quite different in the compound level. So it's we need to do both MS1 and MS2. Again, MS1 alone sometimes is sufficient because we have very high resolution mass spec, and but if we have MS2, we can further improve it. Okay, so this is something you do not have to do MS2, but for us, we do statistics. MS1 is must, MS2 is optional. So this is another concept I hope everybody agree nowadays. You do not need to spend tremendous time trying to identify all the compounds accurately as possible. Approximately correct is sufficient. So you just, if you have a good mass spec, and just the tools can try their best to match, okay? Sometimes it's making mistakes. We call it approximately correct. But if we focus on a function, we talk about the pathways, and they are not just single compound, they are multiple compounds that create functions or pathways, biological processes. They are very robust. So you have a few inaccuracy, doesn't really matter. You can always find the accurate functions. So we did a simulations like 13%, around 30%. Other remaining can be more random errors. You can still identify the accurate pathways. So this is something that just here, you do not have every dot hitting at the center. You just getting around. As long as it's randomly distributed, we will find that functions for you. So this is something we talk about functional analysis direct from peaks. And so it is really no mystery. It's just a well-established statistical concept. So the thing you need to pay attention is that you prepare your input and follow these core considerations. So they need to be high resolution. If you give low resolution, result may not be very high accurate. This is something high resolution mass spec reduce the false positives. So you approximately correct is more than 17% time you get something right? And need complete peaks. So you upload all the peaks you have collected from that your study, not just the significant peaks. And for us, is we think about more than 3,000, is you can get a very good result, because we are thinking if you have a coverage of pathways, we need to have about 300 peaks, something not fully covered, but a significant coverage. So we are talking about 10% is significant, and remaining is a background. So we are talking about 10% of 300 multiplied by 10 is 3,000. So, but usually we have about 5,000 to 8,000, sometimes 10,000 peaks clean after you cleaning. So that's usually enough. But if you just upload 500 peaks, complete peaks, or below 1,000, and it's probably not sufficient. That's just because you do not have enough signal to have this permutation and coverage and get the whole background figured out. So this is a typical output from functional analysis. You have the pathways, and you see the pathways being ranked based on p-values and based on their, on bottom is called enrichment factor. Enrichment factor is basically just tell you, well, did you say it? There's some here. It's what's expected. I see based on what you're doing. This is you hit this pathway basically. Probably two compounds can match this pathway based on the size of the path. But actually you have 8 matches, so the enrichment p-value will be high. So this is enrichment factor, and this is p-values. So somehow this try to help you to to differentiate the both. Like you can think about fold change and p-values. So there's a little bit like that. The magnitude of change and together with statistical significance. And so I'm not going talking too much on this. This is really should be intuitive. And you get the table, you have this interpretation, and you can write your paper. And this is another one we extend on this enrichment analysis, which focus on the significant peaks. And here we just do patterns wherever we see. We just use your mouse to drag it. This is a pattern. This is compounds, peaks, they change together. Want to see which function involved. So you drag and select on the left, and select these patterns and test the functions. So this is something allow you to freely explore and develop your own hypothesis beyond what you define for two conditions. So overall, it is we do this spectral function. From the function, we go to compounds. So, but because it's streamlined and relatively fast, you can keep your momentum and back and forth refine your hypothesis. And at a certain level, you need to purchase the reference standard to validate it. But you do not need to do it at the beginning. You only do it when you develop a lot of good feelings, confidence, and you do it, and that's basically the last experiment before you can publish. So, and this is a summary of what we updated last year. So if you are doing MetaboAnalyst, and you will notice some new changes, and we're actually developing update, and the whole pipeline is updated. And high level is same, but we refine every small details. Hopefully, our Nature protocol will be out in 3 months, and you can see more details, but focus on exposomics. And we have enrichment network. We have better missing value estimation. And so it's thank you for your suggestions. So a lot of changes based on you put on the forum, and sometimes complaints because you didn't do it, and we do feel you, and we have to choose which one is top to implement. But some of them we do see. Yes, I agree. Yes, I would support that features. If you voice your need, and we definitely will consider it. We have a very small team. We cannot do everything, but we do hear you. And sometimes I couldn't sleep well because I know that thing is important. I will do it. So, better graphics, dose response analysis, core analysis. We have a lot of new features. Some is not relevant to some of you, but a lot of time is driven by the whole community, and also my feeling about we need to integrate with the microbiome. We need to integrate with genetics. And especially nowadays, there's a lot of MGIVA studies. We need to do a lot of things. Once we integrate them, it's become very useful. Okay, now here's demo time. I think we should be able to gradually catching up. And
we need to re-enter the Zoom because it's lost the connection just now. So
oh, okay,
yes.
Okay. So hopefully we have the yeah, see, this is same thing. We couldn't join. Let me how about you guys log out? Let me join. And
Can you do? If not, he's on my laptop.
oh, okay, sure. Because once I out, you want to rejoin, and then it's become a thing. Yes. But mine is all set up for the voice. Do you see that? This is something. If you do, yeah.
Because I think the internet is not stable.
Yeah, I know. It's so okay. Well, let's try. Sorry for that, and should be able to set up here. No, that's the HTML.
Okay. Oh.
This should be the other one.
But that will not fit my.
Yeah. No.
This one?
Yeah.
I don't have the adapter.
Yeah, it's coming.
Yeah.
So my computer cannot go online. Her computer is still.
Maybe you use my hotspot.
Well, let's see.
Let's see.
So you can get me online. I can go connect back mine.
Yeah.
But wait, we need to adjust the sound and
Yeah, you can.
first time.
Yeah.
I need to go back to.
Yeah, sure. Yeah, so if you have questions, feel free to ask. We'll wait for the setup.
Jeff, can you hear me?
Yeah, we can hear. Oh, perfect.
Okay. Can I start?
Yeah, I give you a host. You feel free to start. Yeah.
Okay. Here on my screen. So let's start from the slides for the demo. Okay, hello everyone. Welcome to Metabolomics conference, the special section for MetaboAnalyst. I'm in Montreal, so I cannot be there in person, but I'm happy to do this live demo for all of you. So the first live demo session is about LCMS spectral processing. My name is Jianguo. I'm from Dr. Jeff Jia's lab, so I'm also one of the main developer of this module. So I'm happy to do everything for you here. So if you have any questions in the later section, just raise your hand, ask Jeff or ask me. So both are fine. I'm happy to answer any questions. So for the overview of the raw spectral processing, we do have a bunch of workflow, except quite several open source formats, including MZML, MZXML, MZData, and the CDF. Both are open source and commonly used formats for raw spectral data. And from after user uploaded the data, we have the parameter optimization option. And the parameter can be automatically optimized on a pointer, automatically optimized. And with the optimized parameters, we will do the peak profiling, include the peak picking, alignments, and the gap filling. And after that, we will do the peak annotation to annotate all these features into a text or isotope to reduce the redundancy. And for MS1 level, we have the putative compound identification. And for MS2, we have a better chemical identity exploration functionalities. So it's going to be easier for you to explore MS1 and MS2 together. And we also do the result cleaning and the visualization. I will show you the details. And after that, we can connect the result to the downstream analysis pipeline for statistical analysis, biomarker analysis, functional analysis directly. So this makes life easier for everyone. Before I start, I want to focus on several critical points for the lab demo and also for the regular practice. So the first one and the most important one is spectral accentuating. I think Jeff have already introduced in the previous slides about the profile data and the accentuate data. Using the profile data have a lot of redundancy, and the signals are very heavy in the profile data. So we need to do the accentuating first to reduce the size of the files. The most popular one for accentuating is MS Convert in ProteWizard. So this is the one from the Windows platform. This one is only designed for Windows to have their user interface. So if you're using Mac or Linux, you don't have the interface, but it's okay. I will also show you the usage for Mac and Linux. So from here, we can select the raw data file. For example, this one is from thermal, and we specify the output format. And using we have to use the peak picking here. This is the most critical one to add the peak picking as the first filter recommendation, and then we can click start. So it's important to have the peak picking as the first filter. Otherwise, the accentuate cannot be performed effectively. This is a very critical tricky for this software. And in the for Mac and Linux, we have to use a Docker. Docker is a container, including the ProteWizard or MS Convert already in. So from here, you run the Docker pull the image into our local, and we can just simply perform a Docker run to convert the data directly to the open source format. It's the same. We need to specify the picking true, and the level is 1 to 2. So this is usually for advanced users. If you do have a little bit knowledge on Docker, you can feel free to explore. If not, just simply copy paste this command line, change the path, and then run it. You will be fine. So basically, for MetaboAnalyst, we have quite a bunch of modules. Today we are going to cover is the first module for the raw data processing, and we also touch a little bit on this module. This is for MS2 specifically. Usually we have MS1 and MS2 acquired together, so it's better to use this module directly. It's good for us to go from here. For the LCMS spectral data processing preparation, the first step. So we accept the ML, the XML, CDF, and the data formats in the central mode that I have introduced. And for MS2 data, spectra should be acquired in DDA or DDA or source DIA mode for each job. Sorry. Mixed mode is not supported at the current stage. That means if you have both DDA and DIA, you need to submit 2 jobs. We cannot accept DDA and DIA in a single job, so we will support that later. But currently, it's better just to have them separate, different 2 jobs. And it's also not very common to have both modes acquired in a single study. So feel free to run DDA or DIA in 2 jobs. So the required part is the MS1 spectrum. We need to have the MS1 spectrum uploaded as a single individual zip file, 1 zip file per spectrum. Currently, for public tools, we accept 200 spectrum, but we do have Pro Tools. Jeff will give you more introduction later. And we accept DDA or DIA. Maximum is a 50 spectrum. And we highly recommend users to have a metadata uploaded as a plain text file containing 2 columns, spectrum name and group labels. Right now, we are working on this option to allow user upload multiple columns containing multiple metadata factor. For example, I will introduce data later. So one data set could have multiple factors. So we will support that, but we are working on that. It will be there very soon. So you can just feel free to explore after this meeting. Probably in a few days, it will be there. And we also accept QC samples need to be marked as a QC in the metadata file. And the blanks need to be marked as a blank in the metadata file for a blank direction. So basically here, after you upload the data, we will have the algorithm selection. User could specify the parameters. So for example, here you can use the standard wave auto. You don't need to specify any parameters for MS1 data processing. For MS2, there is some parameters related with the instrument. You need to ask the person really run the instrument to get these parameters, these values. We will run everything for you directly. So you don't have to specify any specific algorithm related parameters. So you just need to run some upload your data, specify some specific machine related parameter and run. It's going to be fine. So this is the example. The job has been submitted. I will show you an example later in the real demo. So after the job is finished, we can explore the results interactively. And we also have a bunch of features here. So I have to go quickly to these slides. Finally, we will have a peak abundance table. All features will be listed here. So the feature is displayed as an M over Z value, double underscore, and the retention time, as you can see here. And from here, this is the relative intensities for the peaks. Also, we do support MS2 data only mode. If you have MS2 data only, you can use this module. From here, we support a single text file and MGF file and MSP file from MSL. So you just need to select your data upload. We will process the data, and we can do the compound identification and the visualization of the MS2 matching pattern, also the annotation of fragments. So yeah, this is the specific single spectrum upload. You just need to copy paste everything into this box, specify the precursor and the database you want to use, and iron mode, what's the similarity method. So it's up to you. You can use the dot product or spectrum entropy. Both are fine. The results are similar. It's a little bit different. So after that, you can submit to search, or if you have a bunch of files, you can use the batch processing mode. The data should be in the MGF or MSP file. So when you upload the data, you can just select the specific ions to process. So this is an example. So from the result page, we will show you the detailed matching pattern for the MS2 matching. So as you can see here, this is the top part is the experimental spectrum from your data, and at the bottom is the database reference. So they will be matched together to calculate the similarity. For example, this one is a perfect match. It shows the matching score is a 100. The matching similar score is a 1.0. This is a perfect matching. And we will show you the fragment annotation here also directly. So all the results can be downloaded. So I won't repeat too much on that. So from today's demo, I'm going to show you the IBD datasets. Quickly show you how to upload the data and how the data should look like. From these datasets, we have two samples. The two sample groups. The first group is a Crohn's disease. It's a very typical IBD disease, and a non-IBD control as a control. So a total of 35 samples are included. For this data set, we have only MS1. But when we finish this conference, you can, or later, when it's on the hands-on practice, you can try to play around. But it's better to do this from home because we have so many users in this room. It's going to be very hard to upload the data or make the data processing run simultaneous for everyone. So if you submit your data and it's going to be in queue, definitely, for sure. So because we have so many users, just feel free to explore later when it's in queue, or you can create a URL. I will show you later. So this is the homework. This is also a second example on the MetaboAnalyst website. So it's a whole blood samples, a total of 30 samples. So feel free to explore with the MS2 samples included to see how the data is formatted and how the metadata table is formatted to help you run everything. So after the data processing, we are going to give you the result summary panel, including everything, MS2 result. So it's now this is the hands-on practice, but before that, I will show you do a quick demo on this on how to run the run everything. So let me stop the slides. So let me go to the paper, go to the website. So this is the MetaboAnalyst. You can just use type the MetaboAnalyst.ca and use a different prefix, for example, 3W or new or DAF. So as Jeff had mentioned, we have several domains, but basically you just go to the main site. We will distribute you to the different domains. So there's no difference. They are all the same. Just balance the load user loadings. Make sure everything is smooth for most of us.
Ziqiang, are you showing your screen?
Yeah, I'm still sharing. Okay. Cannot see my screen.
I only see your slides. Don't see your the demo.
Okay, let me reshare. So okay. Share my entire screen. Okay, I think it's good. It's good to know we have the so from here.
Yes, we see it. Yeah.
Okay, good. So from here we have the websites. Let me repeat again. Sorry. We have multiple website domains. So basically, by default, we just go to the main website, which is www.MetaboAnalyst.ca. So from here, we will distribute to the different domains. So everything is same. Just make sure you have a better experience because we have so many users, maybe hundreds of thousands concurrently. So we are trying to just balance user loading to different nodes to make sure everything is fine. So let's get started from here. So basically, it's pretty easy, straightforward. Click here to start. We start from here. This is the module I mentioned for MS1 and MS2 data processing. And this one is for MS2 only. So I won't do the demo on this module. It's pretty straightforward. Feel free to explore later. So for this one, this is a relatively complicated. So this is the LCMS data uploading things. So basically, I have shared the link in the slides with you, so you can find the data from Google Drive. It's something like this. We have all the spectral files zipped individually. So there are going to be a bunch of spectral files. And we also have a metadata file. It's a text file. It can be named as anything, but it must be ended with the .txt. So within this text file, we have two columns. The first column is the file name. It must be exactly match the spectral files. And then the second column is the groups. Here I name it the diagnosis. It doesn't matter for the column name. So you just need to specify the two columns, and then you can upload the data. So from here, you just simply download the whole data set. It's going to be downloaded into your computer. So for example, here, this is my computer. It's a 1 gig of data. It's relatively large. So from here, I just download and extract it. It should be there right away, just a few seconds. So please don't try to do it right now. It's 1 gig of data. If you upload it from quick, so it's going to be very slow. So do it from home later, or you have some time later. So if you upload all these files, it's a 1 gig of files. It's going to be very slow. Uploading will take you 1 hour. So we cannot go through the whole test. So I just, from here, I just show you the basic functionalities. So from here, you need to specify the mode. It's MS1 only, MS1 plus DDA, or MS1 plus DIA. So for this data set, we have MS1 only. So we just specify it here. And go to the data we have downloaded. Here, this is the data we have downloaded. So from here, we can just select all of them. Use Control plus A. So, and then we select open, and all the data files will be displayed here. So after that, you can click upload from the bottom. So for example here, everything will be started, but please don't upload. I won't do the uploading now, because it's going to take at least 20 minutes, even I'm in Canada. So it's better to not to do that. You can try to explore the example. First, it's better to try to explore the quick demo, MS1. This one will be very quick. You don't need to upload. You can see everything similar. This is a very small data set containing 3 groups: health, Crohn's disease, and the QC. This is a very, very small example. Just try to play around with the first example. Or later, you can try to play the third one. This one containing the DDA, it's the MS2. It's going to be better for you to run everything. So I won't show you the data uploading, but I have already submitted a job yesterday. So I will show you the demo on this one directly. So this is the job I have submitted yesterday. So it's already finished. When you finish the job, everything will be here. So for example, this is the job I have finished. And you can see the job status will be 100% finished with all the logs here. It show you everything for the MS1 spectrum have been finished successfully. This is the last night in Montreal time. So when you saw this, you can simply click proceed. It's going to go to the results page, but this step may take a few seconds or even 1 minute, depending on the internet speed and the result size. So, for example, here we have around 7,000 features. So from here we have multiple visualization option. The first one is the PCA visualization. All the features and the samples will be displayed here. So make sure your computer have a GPU, at least a basic GPU. If you don't, it's going to be very slow to rotate this graphics. So for example, my computer, I don't have a specific GPU, so it may take some time to rotate to see the details. And also you can just try to play around. This is the loading plot. This is the score plot of the PCA. Every score is a specific sample. You can double click the dot to see the details for these specific samples. So just for example, here I click this sample dot. It's going to plot a total ion chromatogram for this feature, for this sample. This is a very general summary on this file, and you can just try to click this one to switch the feature into the center. So for example, here each feature is a dot. For example, we have around 7,000. All these 7,000 features are 7,000 dots here. You can just play around. So you can also double click to visualize EIC for each feature across the different samples. So just be patient. I think my computer is very, very busy now, so it's going to take a few seconds to respond. So you can also try to play around from your computer. Hope it's going to be fine. And we also have I think it's the server is very busy now. It's less responding, less responsive. So let's just wait. We also have a bunch of other features, pictures displayed here for you. For example, okay, now the box plot and the EIC here for this feature, and this box plot is can be explored interactively. So you just need to double click a node here, and the EIC for this feature in these samples will be plotted from the right side. So like this one, this is the EIC for this extracted ion from Togon for this feature. It will be plotted here. You can plot multiple one. It will be stacked on the EIC plot. So just make sure. Okay, now they will be all together for this one. So you can also download the picture for reference. So let's go to the second one. This is the statistical of the intensity across all samples and the groups. So we have two groups. Each group are colored with different colors. So, and we have also retention time correction for the retention time correction results. It's also the TIC of all samples, and also the BPI. This is the base peak ion plot, and we have a line BPI. This is the result after the alignment. From the bottom, we have the result summary. We have the spectral sample table. From here, you can just click the icon to see the details in TIC feature. We also have the feature table, for example, here. All the features will be displayed here, and you can also try to view the statistical EICs. And we also provide a basic statistical analysis on these features. For example, the coefficient of variation, the p-values between the two groups, and also the PRs. And we also have the annotation. This is the annotation for the isotope and the adducts. So I think I have run out of time. I'm going to finish this one. Go to the download page. All the results will be here. So you can see we have the MS MetaboAnalyst input clean. This is the clean table with all adducts and isotopes removed. We also have a complete one. It's called the MetaboAnalyst input.csv. So you can use this one for get the complete features for the later statistical analysis, or you can use this one for the input clean. This one is already cleaned without adducts and isotopes. By the way, we are also doing more work on the spectral feature cleaning to make sure we will get a better clean table without just simply removing the adducts and the isotopes based on the interaction and network of the features within the data sets, which will be online in 3 or 4 days, because I'm working on it, which will be done very, very soon. So just keep an eye on the website. So as you know, we are still developing quite a lot for the website, fixing the bugs every day. So make sure we are trying our best to be bug-free. So if you find anything, just let us know. I think that's all there. If you have any questions.
So you have questions, and you can ask our related questions, or you can post on the forum, or, I mean, yeah. Yes.
Basically, you have two files and for like MS1 file and MS2 file.
MS1 is must, MS2 is optional.
But when we have MS1 and MS2 combinedly, then how can we proceed with that?
So Ziqiang, the question is they have MS1 and MS2 combined. How do we upload?
Oh yeah, this is a good question. For this case, you need to use the protein wizard to split the MS1 and MS2 into two files. So after you split the MS1 and MS2, you will be fine to upload because you will have the MS1 and MS2 in two different files. Just use the MS convert to split them. So it's pretty easy, doable. Just an extra step.
So I guess our forum do have probably a post on that, have several screenshots. You need to, when you do the convert, just separate them. Because if you just mixed and sometimes they didn't detect, people complain, so you just convert into separate files. It seems that Protein Wizard can do that. And Ziqiang, if we have a post, yeah, I think if we not, we just create a post. How do we separate the MS1, MS2 when using Protein Wizard? Right? I think you.
Sure, I think it's.
Yeah, you have a post on that. Yeah.
Yeah, I guess I have, but I need to make sure. If don't, I will create one. It's pretty easy and doable.
Can you show a small demo how to separate mixed MS1 and MS2 to separate files?
Probably not right now because we are running out of time. But I'm just saying is separating MS1, MS2 into two separate files should be relatively standard for Protein Wizard. And if we do not have that post created in an omics forum, .ca ourselves, we will create one. But otherwise, it seems.
Yeah, maybe I can quickly show you just.
Oh, you're ready? Okay, good. Yeah. You
just use the subset. This is the MS convert subset. You need to specify the MS1 level. So, for example, here you just type the MS1 level, add it to the view filter condition, and then click start, and the result file will containing only MS1 data. So it's the same. You can just use the two and remove the previous one and add this one. And then click start. Your data will contain the MS2 data only. So it's pretty easy and doable. Just to convert the data twice, you will have a MS1 and MS2 split.
Ziqiang, make sure you create a post on that stuff because I guess people need to remind it. I just think you mentioned it several times. Let's create a post, okay?
Okay, no problem.
Yeah. So yeah.
I have used your this platform Asari to process the data, and in the end, I find several of the HMDB IDs which have been generated in the this data file, but those HMDB IDs are not exist on the HMDB library. So what's the matter with that?
So the question is that they use Asari to do data analysis. In the end, we have some HMDB ID being assigned, but HMDB ID is not in the HMDB current version.
They are
Not existing anywhere.
Oh, okay. It's probably we need to update the HMDB database because the HMDB have phased out a lot of their IDs. So we need to keep the database updated.
So
I think currently we are using the version 4 HMDB. No, I think version 5.
Version 5, yeah. Must be version 5. Yeah.
Catch up the version 6.
So if you have time, just create a post and we'll respond on the post, especially those IDs not there. We want to see, just confirm what's going on. I think we are version 5, but the spectral is more recent, or is version 5? Because we have compound database, we have spectral database, and are they on this? That's where I just think maybe you annotated them, but the ID was old, or what's?
Yeah, the ID probably old because HMDB keep updating their IDs every day. Not every day, it's very frequently. So we need to catch up.
So please follow up on our forum post. We will address that. I get it, but we just hopefully our understanding right. We will confirm when I see what ID is missing. So, thank you for. So we will continue with, so no hands-on. I guess we cannot follow, but we are going to do the demo, and we're still learning. And so next one is microbiome, okay? We go through the heavy lifting for raw spectral processing for metabolomics. If we will focus on statistics, and we have Nature protocol last year, and we are going to publish a new one, so you should be able to all find. But let's just go to microbiome. So microbiome is sequencing based. And how do we sequence the microbiome? There's two main approaches. On the left side, on the right-hand side, is based on marker genes. So we just select the genes. It's called a universal marker. And it's uniquely identified a particular taxonomy level, like bacteria, 16S, and fungi is 18S, ITS. And this is very well accepted in the field. But on the other hand, if you just rely on a universal marker, you may not be able to get too much details. If you're doing a shotgun on the right-hand side, you can get a lot of the genes, and they all help you to pinpoint what exactly species level. Sometimes you can do strain level stuff. So the shotgun is more accurate, but you can imagine if you're doing shotgun for hundreds or thousands of microbes and sequence them, the data will be much more complex. So, but overall, they are both very popular. And the marker gene is so widely used because it's very cheap. I think it's $10, $15. You can get the sample done, and it's really, you can see it. And so this is marker gene for bacterial and archaea, and it's well established. You have this conserved regions. You can use PCR probes or primers, and then the hypervariable regions, they are unique with each species or high level. They can, when you sequence that hypervariable regions, and you can identify their taxonomy level. So it's very genius. You have the conserved to allow you to target, and hypervariable allow you to identify uniqueness. So fungi is slightly more complex. They do not have 16S, but they have 18S. But their taxonomy level is not that very, very deep. And you usually combine 18S and ITS to get to a species level. So fungi is nowadays it became very hot. And a lot of if you watch The Last of Us or stuff, the fungi get to the infections and the pandemic. And here is shotgun metagenomics, and shotgun metagenomics does not care about the markers. They just sequence everything they have in the microbiome community. And in the end, you have everything, all the genes, be it marker, be it not marker genes, you can get a lot. And because it is sequencing everything, you get a lot of information, but the data processing is expensive. Just sequencing itself is expensive. I just said about $10, $15 for marker gene, and this one probably about $300 per sample. And just because you're sequencing a lot, you want to sequence deep and broad enough to have the good coverage. In that case, you get a lot of data and sequencing reagent cost. So it's usually more than several $100s. I know the beginning is $1,000. Now it's reduced, but still it's more expensive. Now we need to process this sequencing data. I mentioned the sequencing processing such as RNA-seq is so well established. So RNA-seq is not a problem, but marker gene And shotgun sequencing is different from just RNA-seq because it's so we're talking about several tools. One is called DADA2 is for a marker gene. It's a single marker, be it 16S or 18S ITS. This is already available in MicrobiomeAnalyst. Then there's some other tools called MetaFlan. If you're doing a microbiome, MetaFlan is very efficient. It's a command line Python-based tools to use multiple markers to more accurately assign this taxonomy. Then you want to get the functions, and there's another tool called Human, which is designed and developed during Human Microbiome project, and working with MetaFlan to get functions. So we're doing a lot of microbiome studies, and we realize that's quite inconvenient because both of the tools, you require depending on a lot of things, and you need to do it separately. One get taxonomy, one get a function, and can we do things simultaneously, faster, all in one tools? So this is sometimes I was really hope we have such tools. And this one called the FunTaxSeq, and it was supposed to release by now, but there's some delay, so we hope release it later this summer. And hopefully everything is all in one, fast, and you can do it through online tools or download doing a command line. So if we do a marker gene, and how do we do the data analysis? And so we need to convert the sequence reads to assignment called ASV. ASV is amplicon sequence variant. So ASV is a unique piece of sequence that can identify this microbes. Let's see that. So there's some other things called a phylotype or OTU. I'm not talking too much on that just because it's more or less not so current. The current one is ASV. So ASV is a variant, and it's just sequence itself, about 1 or 200 base pairs, and it's very unique. And once you identify that, and this one you can almost compare across. And if they're really, you have some close hit, you can actually annotate them. So this is something very innovative in the last several years before, because before we are talking about OTU. OTU is operational taxonomy unit, and you want to cluster the sequence that are highly similar together, and class together, then you assign a unit. And it seems intuitive, but practically it's very computational intensive, and also it's data specific, because every time you clustering and use the class to identify a unit, it's really based on specific your data at the time point. And it's very hard to compare across different studies. And then people are saying, why we do the clustering? We just directly, uniquely identify the sequence itself, and which is comparable, which is universal, and you do not need to perform clustering, and so it's efficient. And indeed, and it's making the field is much faster and easier. So this is gradually people use the ASV, and this is also what we are supporting. We do not support OTU, and just because ASV is much better. Now, this is the next one. MetaFlan was based on the multiple marker genes. Before we talk about 16S, 18S is single. But if we're doing a shotgun, we do have multiple marker genes. And they are not universal, but they are at every lower level, they have this capacity to help you refine the signature. For example, here you can see this a green, it's very unique, defined as clade. They call it clade specific marker. So they are not universal, but at certain branches, it became very unique. And you can actually combine them to help you refine the assignment of this different taxonomy level. And this is exactly what MetaFlan was doing. And they create such a huge marker library at a different level. And you can see it's millions and close to species level, clade specific. So this is making a lot of sense. I guess the most challenging part is create such a high quality library. But they are well maintained, they are doing well, and they are fast because they are not talking about every sequence. They just focus on these multiple markers to do the assignment. So this is very good. I just talked about the human pipeline. This is developed together with MetaFlan to do the functional analysis. What they try to do is that you have the shotgun. They're doing this orthologs mapping to these enzymes and create the pathways. And this pathway is based on CAG, so it's based on metabolic pathways to get functions. So overall, this is a high level talking, but exactly what they're doing is slightly more advanced, complicated. And you can use a pipeline to get functions. So again, it's, I think they have a lot of dependencies, and you need to use it to get functions. What we would like to do is we want to get simultaneously get the taxonomy and the functions, because you get these reads and you get markers. You get some of them belong to some of us actually have the functions. So we should be able to do it all together without doing it twice. And this is actually already done, but we need to do benchmarking. We want to make sure their performance is similar or better, and also save enough computing power. And this is done, I believe, in C++. Yao is C++ or C? C++. Okay. So Yao is we are doing benchmarking compared to other alternatives. We want to make sure it's fast and get everything and also functions. So this is some of the ideas behind, not some of the design behind. So we do the DNA to DNA mapping and try to identify as much as possible. This is exact. If we cannot identify them, we do translate to DNA to protein. But once you go to DNA to protein, you have more chance evolutionary distance tolerate. So we try to mapping as much as possible. Then we, those finally you cannot map, then we will discard, because this is where they get, hopefully get enough. Then we will get a taxonomy table, we get a gene abundance table, and ASV table, and we'll upload to MicrobiomeAnalyst and do a functional analysis. So you can imagine this one is part of the, going to be part of the, just like a spectral process, you see it. You upload your marker gene, you upload shotgun, you will get it done using the server, and you do the statistical analysis and continue on. So this is going to be part of MicrobiomeAnalyst once it's in, let's say, three months from now. And the database is important. So we create almost 17 million genes and proteins from 18 prokaryotic. And we also use this taxonomy trees and to really make it as high resolution as possible. So this is a FunTaxSeq website. We are still working on benchmarking and documentation. So far, it's all functioning, but it's a lot of things we need to more accurate if we're aiming to release to the public. So if you are adventurous, you feel free to try. But on the other hand, wait for 3 months should be better. So this is the output from FunTaxSeq and raw data and tables and the functions. So this is exactly if you need to run multiple tools using Matplan or Kraken or Human, but you just run this one single tools and you will get all the result. And most importantly, you do not need to run this command line. And you run it within MicrobiomeAnalyst is a graphical interface. And because it's written in C++, actually quite efficient with a very good memory and the CPU. The reason why we emphasize performance, we're talking about hundreds of thousands of users at the same time. So we have to be very cautious. And so, yeah, that's about the raw data processing. So what the coming is how do we do marker gene data processing using DA2. So it's already in the tools. FunTaxSeq, we are going to add maybe 2 or 3 months later. And yeah, it's your turn. Okay.
So next, I will demo you on how to processing the 16S sequencing data using our online tool. So you can see we have a raw data processing module embedded in microbiome.
You're not on the presentation mode.
Oh, okay. We have the raw data processing module in MicrobiomeAnalyst. So you just click here and to do the processing. So next I will just quickly introduce the raw data processing workflow. So basically we use commonly used very efficient method, the DADA tool, like Professor Xia mentioned before. And we got the first step is raw data input, and then we have a sanity check to control the quality. And then the important step is that we need to tune the parameters for control the quality of the sequence, and then we will processing to finally get the taxonomy annotation and the abundance table, ASV abundance table. This is the two main information we can get from the marker gene sequence. After we getting those tables, we can then do the microbiome analysis. So in this module, I will basically show you how to get those tables. So here we use IBD example data. So it's from the human microbiome project, part of the project, and we use a subset like 35 files, which were generated using the Illumina platform. It's showing here. They actually use a pair-end method for generating the data, but their online database only provides the the forward sequence. So in this example, we will use the single end data, but it doesn't matter. All the online processing, it's keep the same. So I will show the web page for processing. Here, you can select the data you want to processing to upload. I will keep this working because it takes some time as the connection is not fast. So here we show the example data. You can download the data like using this link, and we have 35 FastQ file. Usually we require you compress a file. You cannot directly use a FastQ file. You need to compress it as the fast.gz or zip the file, both okay. And then very importantly is that you need to name your files end with the /R1 or R2 to let our tool recognize it's the forward sequence or the backward sequence. And we also require a meta file, so it looks like this. You can have several columns for the metadata information, but the first the sample column the name should be exactly same as your sequencing data. And let me check, it still takes some time. For the public version tool, we accept at most 100 files once, and each file it's like at a maximum 100 megabytes. But if you want to handle larger or more files, you may use our pro version tool, or you can upload several times, because DADA2 have inherent pipeline that can handle the batch effect, so you can combine the data actually after processing. So after you upload your files, and then you just click proceed. And there will pop-up dialog to check if it's a single-ended or pair-ended. And if it's all right, you can just click yes to go to the next step. Usually, if the connection is good, it takes like much less time for the uploading and processing these steps. So here, this is also important for control your next step analysis. And here you can see the first column is the data file you upload, and then you see the reads. When I download the raw reads from the database, actually here you can see some of the sequence have really low quality, like the reads totally is like less than 100. So I remove those samples. So here you can also control the quality here to remove those like really low quality sequencing data. And here we have a validated column which show if the FASTQ file is well formatted or it's validated. So you can only pass to go to the next step if all the samples are validated. So here shows the group because we use the IBD study, so we have two groups for comparison. This will explain later in the statistical module. So here all the data is like all right, so we proceed to the next step.
Is it delete is on this page or
no? No, there's not a delete. Actually, we can add one.
The last column delete.
So here we got the parameter setting page. This is also important because like this, in this example, we can actually see the quality of the sequence is relatively good. You see it's like a deep sequencing, and then from the start or the end, there's not too much noise. The slope, actually usually the quality score, if it's up than 20, we can totally acceptable. So here it's all up 30. So we can even don't cut it, but if you like want to control more quality, you can cut the from the first, like for example the first 25 reads and the last like 10 reads. So here we have the sequence trunk parameters. We have the trunk length and the left trim and right trim. Actually, these three parameters in DADA2, it will like firstly processing the trim left and then the trunk length. So usually we directly set the trim right is 0 because it's no use. So here we can trunk from the first 20 like 24, 25, it's both okay. And then we control this to, like, let me see, 220. And then, which means we cut the final, like, 15 reads. And here we also got other parameters, for example, the max error expected, which means we cannot, if the sequence have more than 2 expected errors, then it will be removed. And also there's other sequence, the parameters, maybe you can read more from our tutorial. The most important ones are the max error and the trunk length. And then we need to select the sequence type because we use the 16S, so we just select this. And another thing is the taxonomy annotation. For the 16S, we have 3 latest version database, which are also widely used for the Silva and RDP and the green genes. So in this example, we just select Silva, and then we can submit to processing. So So this may take some time, so I do a local version. So this is a result, what we can get. So when the processing, you can definitely see each step. Usually the first like 70% processing will be fast. And actually the slowest step is the taxonomy assignment. It takes a long time. Not that long, but it's you need to wait a little with patient. And then we proceed. It's a result page. We got 3 tabs. You can see first library size view for, we got the library size for each sample. It's their distribution. Then you can see how the sample was processed in each step of our processing. You got the input length, and then we filtered, and then denoised, and then finally we tabled the ones that remove the tumors. And here we also got the taxonomy annotation. So this is the final ASV sequence we have got for each sample, and then the annotation was listed here. So you can see this. Usually 16S, it's annotated to genus level. This sequencing data actually deep, so we got quite a lot of species annotated here. Then we can go to the final page to download those files. Here we can see these files input can directly go to the statistical analysis. We prepared the sequencing abundance table here, and the metadata here, and then the taxonomy annotation here. So these 3 data will be directly used in the marker gene and statistical analysis module, which we will have a demo in the next section. So I think this is the raw data processing for the marker gene sequencing. Do you have any questions?
So actually curious, how many of you have done microbiome or intend to do microbiome data in, like, say, in coming years? Can you raise your hand? Okay, not so many. And for me, is I do a lot of metabolomics, but I realized that you have to integrate several other omics technology to understand biology. So multi-omics, and it's very common. And I'm actually in a microbiology department, so people doing microbiome metabolomics very common. So, so I just hope some of you will eventually use more than one omics to do data analysis. And today we just showcase the microbiome metabolomics, but we do have other in multi-omics integration. So, and the next one we are going to focus on is actually quite different compared to what we usually do with metabolomics statistical analysis. Microbiome data is not just a data matrix doing stats. They also have some ecology concept. So there's some concept we need to pay attention to before we do data analysis. And while we are close to 2, I'm just not sure. Are you okay? I'm continue, and then we are going to do a maybe demo hands-on, and you're free to take a break. Is that okay? And so yeah, because we have 4 hours, so at the moment is they have a break 15, but my suggestion, let me finish and we have a break. Is that okay? Yeah. Okay. I'm not suggesting a break. I'm just suggesting I'm finished and we have a demo and break time. So that's stuff. So here's an overall view about raw data processing. Yao already did a demo. So from raw data processing, we are going to have a taxonomy table and ATO ASV table. So once we have these two tables, we can actually do the statistical analysis. If we do shotgun, we are going to have another table called a gene abundance table. Then we can do shotgun functional analysis. So overall, is I mentioned earlier, the raw data processing is more or less standardized. You can use established tools, including our tools, to get that. But now it's stats profiling is taking your time because nobody else can do it for you except yourself. And otherwise, that's what's the fun of doing research, understand biology. So this part is that we need to pay attention to, and I just have more slides on how to do a statistical analysis, how to do a community profiling. So this is not available for metabolomics because metabolites is more or less just a single layer. But for microbiome, they have actually different layers. And so one thing is, let's see how many species there, how many taxonomy there. It's an overview, visualization. Then we need to talk about alpha diversity. Then we need to talk about beta diversity. This part of the thing is new, okay? For metabolomics, we do not talk about this. So I'm just bringing to your attention. And this is very unique, and so we need to pay attention. So one thing is called a rarefaction curve. So we do a sequencing. We sequence more, we get a new species, right? And so you do not want sequencing too much because you get in all, when you reach the plateau, you're sequencing more, not necessarily getting new species. So their rarefaction curve allow you to decide at what sequencing depths you can stop, but maximize your discovery, but save you money. So this is something sometime within the tools, the rarefaction curve, and you need to be comfortable with that. And this is a stacked bar area plot. It's very commonly used in microbiome data. And just use PCA for metabolomics. So this is very common for microbiome. What it visualizes is the composition for each individual sample at different taxonomy level. For example, here you can visualize the species or family levels, and the different color represent different proportion. Okay, this is in this case it's a relative proportion, and all added up to 100 or 1. Okay, this is just to visualize, and if you sort them, sometimes you can see some patterns. Oh, this have high proportion of certain bacterial taxonomy. So it's very intuitive and visually also very appealing. So you should be comfortable with this. If you do microbiome, this is something is quite commonly seen, and it's relatively easy to generate using our tools. And this is one you can actually not just directly view, you can actually quantify them. So this one is actually, so how many species in each sample? And you can do a little bit error bar because sometimes you see very, you can do a little bit permutation or bootstrapping and see what's you can estimate. So you can see the individual number of species. And all these species is a total number observed. They are not necessarily shared, okay? They just diversity composition. So this is one. You can also test a whole group whether they are significantly different or not using like ANOVA stuff. So there's a theory, and most times seems true, is that diversity, more diverse, is more healthy, okay? And IBD or certain disease lose the diversity, and so they are dysregulated. And like, so more diverse looks good. So healthy people are usually more diverse than disease. So when you have disease, conditions become more stringent and reduce the diversity. There's always different theory, but this is diversity, alpha diversity. So alpha diversity I showed you before here is CHOW1 is mainly observed. So you have a little bit of flexibility to see the actually observed, then a little bit adjustment. Then there's we need to think about diversity. It's not just total number of species. There's something else called evenness. So for here, you totally have about 5 different species, let's see. But here it's also total 5. the consequence is different. Evenly distributed versus very type dominant species. And function-wise is also different. So sometimes they want to balance them. They want to have a mirror that capture both diversity, both richness and evenness. So there's called the Shannon diversity, the Simpson diversity, slightly emphasize different part. And so you just need to pay attention to ecology concept in the microbiome. There's several names, terms, and you need to be aware of. And so in our tools, we actually have all this kind of thing. You can see the Chow-Wan, ACE, Shannon, Simpson, and Fisher, all of things are there. So you can explore, but when you write your manuscript, you need to make sure you understand it right. And sometimes I'm not just overload you right now, but you just let you know there's different mirror, and they emphasize different things, and you can explore which will make most sense at the moment. Try to learn, oh, this Shannon diversity is much better than Chow-Wan in my case, and because Shannon is more emphasized richness and Simpson is more evenness. So there's at that moment you learn, you will feel much more meaningful, not necessarily just right now. So you can test whether they are significant. So we talk about alpha diversity, which is just care about the total number of species, sometimes talking about evenness. This species may not be shared across samples, okay? They are not trying to align them, so you're just talking about that number. And what's more meaningful, people want to identify those shared, the core microbiome, people shared across the population, a cohort, and this is more meaningful beta diversity. So this is, they use different name, alpha diversity and beta diversity. So just, if this is new to you, first time, just go with me, and next time you will be more comfortable, okay? And beta diversity, we have G-card index, we have Bray-Curtis distance, and there's another one called UniFrac. So UniFrac is considering some of the phylogenetic distance, so it's probably sometimes more meaningful. So all of this beta diversity mirror give you some output and do look like a PCA, PCOA. And here's, I'm just talking about in our tools, we have all these different kind of measurement and allow you to visualize them and test them and also test their significance. So this is when you will be, hey, it looks like PCA, but they do not call it PCA, they call it PCOA, it's principal coordinate analysis. So you can actually see they are actually similar interpretation. Because the distance is not Euclidean distance. The distance is ecological distance, like the one I just mentioned, and UniFrac, and all this Bray-Curtis and J-card, all this weighted and unweighted. So all of this is there's no best method. Okay, it's just you need to explore, find which one is meaningful for your data, and test whether they are significant. And here's what's generated in our tools. On the left side is traditional beta diversity PCOA plot. And you can see different conditions, actually, separating very well. And just like it looks very exciting because you know they are very coherent, the disease control. And this is actually probably different age group, young, old stuff. Now, you can actually combine them. You see this is alpha diversity, the color corresponding to the alpha diversity in each individual sample. We already know the alpha diversity here. You see this? This individual sample, they have different, we can actually color them right on top of this PCOA plot. And you can actually see the individual diversity is also drive, but this is really integrating this to find out what's the main factors. So it's give you some insight and allow you to actually find out which is the most important factors. And this is another one called HG. I do not know, we have similar in Metabolomics. And it's allow you to see the whole distribution from the phylum all the way to species level. Because this is hierarchy. For metabolomics, we just see the peaks or compounds all flat, same level. But here, they allow you to see from phylum all the way to species. And you can see at what level is most different. And so this is a bird's eye pictures from everything. And you can compare the conditions and subtract them and see the different colors at where it's most different. And some people actually do this time series of different treatment at different point. So on the left side, you have a tree and just labeling what it is. On the right, on this side, it is actually shows different point. And this is generated using our tools. You can actually label, unlabel, and each groups use color and gradually build a storyboard. Oh, this is the stuff from low to high, or from enriched to depleted. So you can actually build it. And some people are really creative. They use the visualization and even statistically do not have tools to evaluate, but they can actually use them visually build a story, and very interesting. So before we talk about unique to microbiome, alpha and beta diversity, now we need to do some statistical analysis. And microbiome data is more or less have several, compared to metabolomics, they are sparse, containing too many zeros. So this is not usually present in metabolomics, okay? They have vast difference in sequencing depths. And sometimes it's more than 10 times different. It's really distributed, and the differences in different shapes almost. And you also call it over-dispersion. You think about this normal distribution. No, it's just so spreading out. And so most time at the beginning, people always use non-parametric test because it's not normal and it's no way to bring it normal. So everybody uses non-parametric, but non-parametric seems not so powerful, and people want to develop something more unique to take advantage of the distribution. So there's a lot of development in the last, in the first 10 years or 5 years during microbiome study, and that was quite a time and which I was very interested to see how people are addressing that challenge. And during the time, and one of the key part is normalization. So if we filter and normalize the data, we can reduce or control the dispersion or difference and make it more comparable. So normalization is very important for statistical analysis. It's not so meaningful if you're doing ecology analysis. You're just doing alpha and beta. That's we already covered. But here we talk about we want to compare samples. We want to see biomarkers. Now you need to normalize because you want to compare things that are meaningfully comparable. So you need to normalize. So this is when you spend a lot of time in MetaboAnalyst. Same thing in MicrobiomeAnalyst. Normalization is very important if you want to do comparative analysis. So we talk about rarefying. I just talk about rarefying is we just subsampling the data. If you are so deep and the other one is shallow, you want to compare deep sequencing versus shallow sequencing, the difference is not so meaningful because the difference is just because the sequence is more, right? So we need to do that, called rarefying is actually subsampling the deep sequencing to have the similar similar library size, okay? Then you do scaling and you do transformation. This is similar to MetaboAnalyst. You have statistical things, but overall, it's a critical step. We do not know which one always working the best, but this is common approaches. Now, here's a comparative analysis. We normalize data. We want to compare them. Which the method is most useful? And it turned out, and there's in the middle is microbiome specific. A lot of publication is on the Nature Method, Nature Biotechnology, saying we found a distribution of microbiome, and this is unique for microbiome. It's really good, and people are just so excited, and try to see whether it is the case. But when the dust settled, and people found out a lot of the just the RNA-seq method, like EdgeR-D-seq, and actually doing very well. It's comparable. So a lot of things you see it uniquely. Microbiome is not necessarily always working well. So generalized method like EdgeR-D-seq, which is designed for RNA-seq, actually doing very well. So how many of you doing RNA-seq before? Yeah, you familiar with RNA-seq? Yeah, some of you. Yeah. So I guess microbiome and RNA-seq is really slightly, I think, is a bigger field, and it's probably a much larger than metabolomics. So they have a lot of algorithm developed. Some of them is quite useful. So here's single factor differential analysis, and we try to leverage the different phylum, the color and the background. You can use a drop down of the method. You can see the AGR, t-test, ANOVA. So we give a variety of things. The reason is that we do not believe there's a single best method always working well, and so allow you to explore. So this part is that unified, defined. We are not restricting. We just providing and give you some guidance, just because we a lot of time is specific to your data. So this is one that added relatively recently called Maslin, and it's based on the linear modeling. So we actually found out linear modeling is very good compared to t-test. Just so linear modeling is more flexible. Linear modeling can consider a lot of covariant. Basically, if it's doing population studies, a lot of the other factors rather than experimental condition. For example, you want to control age, gender, and BMI or different things, and linear modeling can allow you to control that. But if you're doing t-test ANOVA, it's very hard. So you can see when you do this stuff, you can control different factors while doing a statistical analysis. So this is more suitable when you have a complex study design. And this is another thing that actually drive me to microbiome study. At the beginning, I showed you that taxonomy and metabolic function is very different. Here it is again from another visualization. You can see there are a lot of difference in the phylum. Phylum level is very high. But when you do their metabolic, like COG, this is metabolic enzymes pathways, it's very flat, it's very stable. So people think about a lot of redundancy. Disregarding you have different microbial composition. The function is stable, which making a lot of sense. So where is the function? How do we measure the function? This is where metabolomics helps. And we can actually predict function from the marker genes. We can predict function from the shotgun. So clearly, we can measure using metabolomics. And where the functions, and there's several functions called predict functions. So we know, we measure them. We know if we have their whole genome sequence or we have their closely related genome, we can use that to predict, this microbe, they have this enzyme, they probably have these functions. So using their genome sequencing or closely related genome sequencing to predict the function. So this is one of the most widely used tools called PyCrust, and they use a CAG database and based on copies and predict the functions. So it is all available in MicrobiomeAnalyst. So you upload your marker gene, you can do functions, you can do stats, alpha and beta diversity analysis, you can do functional prediction. So, and I think now is time. We will do demo. Also, you can take a break. And we can, I think, what's the time to come back? It's, let me see. Hands on. Next one is 3. So yeah, we have some time to explore. So this is part. We also have a break. So all of this section is yeah. So yeah, you.